Okay, I now have the honor of introducing our guest speaker from Australia, Dr. James Elder, who will talk to us on the genetics of familial exudative vitreoretinopathy and Norrie's disease. He's done a lot of work in pediatric ophthalmology and its genetics. So let me have the pleasure again of introducing to you Dr. James Elder.
Thank you. I'm very honored to be here and somewhat humbled, actually, because I'm not really a geneticist, as most of my panels are. I'm a clinician, and this is a clinician's input on these groups of disorders. So first, a little bit of history. Norrie's disease, in retrospect, was first described by Gordon Norrie, who was a Danish ophthalmologist of Scottish descent, which explains why he's got a very Anglo-Saxon sounding name, but worked in Denmark. And he described a group of boys with congenital visual loss who, by the time he saw them, really had thusical eyes. It was Mette Warburg in the 60s who defined this condition more accurately and gave it the name Norrie disease in honour of Gordon Norrie. And in the mid-60s, she recognised that there was gross abnormalities with the development of the retina and retinal vessels, with proliferation of the retinal vessels. Creswick and Scapins in the late 60s described what we now all recognise as familial exudative vitreoretinopathy with exudative and tractional changes in the retina, and gave the name to this condition that we now recognise. And this condition, to clinicians here, will be well recognisable. In more severe cases, you can have total retinal detachments or retinal folds. In less severe cases, you have incomplete development of the retinal vascularization, and what, if you were looking at a premature baby, you would say is peripheral avascular retina. And these less severe abnormalities, if they're just observed, can sometimes progress, and so you can go from having an avascularized retina to a retina with tractional changes over time if you don't intervene. Prendergast and Tracy classified the various degrees and severities of the retinal changes in this condition. So, broadly, we recognise that there are phenotypes that are purely ocular, and the mutation in the Frizzle4 gene is an example of that. It's autosomal dominant, and it only has ocular features. And the archetype, or the typical prototype familial exudative vitreoretinopathy, which has systemic disorder, is Norrie's disease. And so some people with the Norrie's mutation will only have fever, others have systemic abnormalities, including hearing loss, intellectual disability, dementia, and behavioural problems, to name a few. The Canadian Fever Consortium published a paper recently, earlier this year, in which they looked at the genes that are associated with fever. The ones marked with a red star are part of the Norrin-Wnt signalling system, so that's Norrie's disease, and some of the fever-associated phenotypes with the other different genes. And then there's a whole host of other genes, and this is in fact even an incomplete list. I can think of a couple of conditions that are not on this list. Revesz syndrome is an example. And what this group suggested is that when you're doing sequencing, you should do a sort of a targeted approach. Start with mutation analysis, looking at things that are in the Norrin pathway, and then go a bit wider. And some of these are also recognised on the basis of the syndromes. Before going on to consider a little bit, I need to consider some of the development of the inner retinal vascular system. Basically, this is a neuronal advancement from the optic nerve that drags the blood vessels behind it. And so a lot of the factors that are important for the subsequent development and angiogenesis are actually derived from neurons, the platelet-derived growth factors, VEGF, and importantly Norrin, which comes from Muller cells. This is a fairly old schema of the bits and pieces that are involved in driving vascular development in retina, and the Norries pathway is on your extreme right, and it's interesting that exactly how it interfaces with this is somewhat uncertain, and there are a whole lot of other presumably undiscovered genes here, and I'll talk about that a little bit more. So the Norrin-Wnt signalling pathway is a very important pathway in embryogenesis, and Norrin is a protein in the retina that's secreted by the Mueller cells. It interacts with a number of receptors on the cell surface, and they stabilise a intracellular protein B-catenin, which then passes into the nucleus and causes gene transcription. And I'll talk about the association of the severity of phenotype with different variations in this. Just to digress for a little bit to talk about Wnt signalling, this was first described in fruit flies, so these are very important secreted growth factors that are involved in embryonic development in all animal species and in most structures. The interesting thing here I discovered is that the original description of the wingless gene was here in New Delhi in the Institute of Agricultural Research a little over 50 years ago. R.P. Sharma was the scientist that described this, and this was the beginning of our understanding of this very important embryological pathway. So, in terms of genotype-phenotype correlation, in Norrie's disease, with the abnormalities in the Norrie disease protein, this is getting better understood. There was quite a nice meta-analysis published a couple of years ago now that looked at the published literature and in terms of the ocular phenotype divided it into mild, which would be peripheral avascular retina, moderate, in which there are significant exudative changes and the beginning of some tractional changes, and severe, in which there was total retinal detachment. Here's some examples of moderate and severe abnormalities. And what they showed is that there is a correlation between the severity of the retinal phenotype and indeed some of the systemic features, particularly hearing loss and intellectual disability, depending on what part of the Norrin protein is affected. And look, it's quite a detailed paper, but there are a couple of things. So if the effect is in the more peripheral binding site with the LRP5 receptor, then you don't get severe retinal phenotype. Whereas if it's in the cysteine, which is very important for the formation of this cysteine mutation, very important for the formation of the Norrin dimer, then you fairly universally get a severe retinal phenotype. So there is some emerging understanding of that. And the same thing applies, and this is just briefly, again in the phenotype with the associated features. Again, the peripheral part of it, of the binding, if it's with the LRP5, there are no systemic features, and so you tend to get, that's when you get the fever isolated pattern without systemic features. And it's a little bit variable with the others, you can see with the cysteine, rather than them all having significant systemic features, a significant number do. And so there are these emerging understandings of that. Now, where are all these missing genes? They may well be in the other parts of what I'll call the angiogenesis cascade or control systems, but Professor Sundran presented a paper in his presentation on enhancers and regulatory elements, and I think in fact that's probably where a lot of the remaining genetic discoveries with this condition needs to be. And one potential example is if you look in OMIM, exudative vitreoretinopathy type 3 doesn't have a gene associated with it, but its locus is very close to exudative vitreoretinopathy 1, which is the frizzled gene thing. So I do wonder as a clinician whether someone's going to identify a regulatory element there that controls the frizzled gene. And this is important. And the other example is nothing to do with fever, but I was recently involved in a paper with some colleagues from the US in looking at the cause of hereditary congenital facial paresis. And this is actually caused by a regulatory element that silences the GATA2 gene And so, we are going to see more and more of this over time. And these regulatory elements are in the non-coding area. The junk DNA is perhaps not quite as much junk as we thought. I wanted to talk very briefly about treatments for these conditions. So in some respects, they're a little bit like severe retinopathy of prematurity. This is a child I saw with Norrie's disease who was born at 34 weeks, not because we knew this pregnancy was affected. The mother didn't seek antenatal advice. So am I allowed to go for a little bit longer? Yes?
He's asking something.
Yeah, it's got a time
Take
up.
time, please, take your time.
Yeah,
Very
right.
important.
Yeah. So in this condition, this child was born at 34 weeks, and you really couldn't see much of the posterior pole because of extensive hemorrhage, but the peripheral retina did not appear to be vascularized, and it was a very low quality fluorescein angiogram we got because of the hemorrhage, but there were no retinal vessels beyond where the hemorrhage was. So this child was treated with bevacizumab, a VEGF inhibitor, and 4 weeks later, this is what we saw. The retinal hemorrhage had largely cleared. You could see that it was a very immature retinal vasculature with retinal vessels not really even getting to the fovea. And this is an OCT done at the stage that showed that there's gross perturbation of retinal development. You can see inner retinal layers, but they cease at the fovea, and it's just sort of unrecognisable as a normally developed retina, and this is at 38 weeks of gestational age. So the child then had extensive peripheral laser. This was just taken immediately after the laser, so you can see edematous retina. But all of this was to no avail, because this child had an examination under anaesthetic a couple of weeks ago, 8 years on, and this eye is NPL, the other eye is thicicle, so we were unable to arrest the process with this thing, and that's because there's so much disturbance of the angiogenesis pathways, and so even though the child had appropriate treatment, there are a few descriptions of this treatment being successful, but were probably with less severe mutations than this child had. So where are the other therapies going? From mouse studies, we know that if you have an ectopic source of norrin, so if the Norrie's protein, so if you have a knockout model in which norrin is not produced in the retina, but you engineer the mouse to have an ectopic source of norrin in the lens, the retina develops normally. So if we had some way of giving the child a supply of norrin, but it's not